I think that before I talk about endocrine disruptors, I will have to talk a little bit about what the endocrine system is. And now, okay, see if I can, it seems that I cannot move this. So first technical problem, okay. Wonderful, thank you very much. So I will start from the very beginning. And so we are talking about the endocrine system. We say that the endocrine system is the group of cells and organs that produce chemical signals called hormones. They travel, those hormones, through the bloodstream in order to reach their respective target cells. So here you have the whole complex system. The ones that you would see more often when referring to plastics, for example, are endocrine disruptors that mimic one ovarian hormone, estrogen, and then chemicals that interfere with the action of the thyroid gland. But what I want here is to show you the different types of effects of endocrine organs so that we best understand what to expect about endocrine disruptors when people are exposed to them. So as I said, those are very powerful messengers that are present in the bloodstream at very low concentrations. Some of them 10 to the minus 14, 10 to the minus 10, you name it. Some of them 10 to the minus 6. And they are acting exclusively upon their target cells. So those target cells contain receptors, but that is not just the most important part of the story, because since we discovered discovery of these receptors, the concept of receptor, it's at the middle of the 20th century, and then the characterization, molecular characterization of these receptors happened in the decade of the 80s, 1980s. So we know that some of them are located in the plasma membrane, so they bind, the hormone binds the receptor, and then through a very complex set of a cascade of interactions, there is a second messenger that will travel to the nucleus where these hormones will change the pattern of transcription. Then we have nuclear receptors that are the ones that, for example, estrogen receptors are located in the nucleus, and estrogens that are very liposoluble will cross the capillary membrane, cross the cell membrane, and go directly to the nucleus, where they will also affect gene expression. Those interactions are very complex, and sometimes, like if you precipitate with an antibody or do a chip-to-chip, etc., you will find like 150 proteins that come down with your receptor. So it's a whole network of proteins that is impinging on the effect of those hormones. It's very complex, and this is just the first step. And the story doesn't end there, because we shouldn't be reductionists and only look at that. For example, today, we do not know actually how is that thyroid hormones have this very striking effect on the developing brain from myelinization, etc. We know that is happening. We know how tadpole converts through metamorphosis into a frog. We can observe that. We do not know all the processes. Okay, so this is just the tip of the iceberg, and even at that point, the ligand receptor DNA interaction is extremely complex, and that is what most people were trying to elucidate in the 30 years or so since we started working with those molecularly characterized receptors. But what is important of the endocrine system is their functions, because those hormones have a role of maintaining the constancy of the extracellular fluids, and that we call homeostasis. And for example, insulin is very important on the regulation of the glucose concentration in blood. Why? Because also these hormones that act on metabolism control the supply of nutrients into the cells of the body. For example, in diabetes, there is an impaired utilization of glucose in muscle and of amino acids as well. And then other very important regulatory endpoints are in development during morphogenesis, growth, and reproduction. So hormones are like the second nervous system, although it works only at distance, but it's as important as the nervous system, maintaining and provoking, if you will, the development of an organism. So we can also say that endocrine functions are on the organizational end, and by that I mean hormones will affect the developing organism, and in the case of thyroid hormones, we need that for fetal brain development, and without that, we lose IQ, okay? So we need it to achieve intellectual capacities, cognitive capacities, also for myelinization. So you see kids, when they try to tie their shoes, they cannot do it correctly until they are about 4.5 years of age, because at that point is when myelinization is completed. And all that is under the control of thyroid hormone. So those are called organizational effects because they are irreversible. Either you have the hormone at the right time or you lost it, okay? It's morphogenesis that is being affected. And then we have what is called activational effects that are reversible for the most part. And so they depend directly on the interaction of ligand and hormone throughout life. Like, for example, I can run an experiment in a rat. I ovarectomize the rat, the lining of the uterus shrinks. So now I administer estrogens, and the lining of the uterus grows, and I remove the estrogens, and it atrophies, and back and forth. So we call them just activational effects. But remember the organizational effects, because those are the most striking ones. So Howard Byrne, a very important person in endocrinology, he was one of these pioneers of comparative endocrinology, coined the term the fragile fetus. And indeed, here we have receptors. For example, estrogen receptors are already present here. This is the zygote. Oocyte, right after fertilization, it divides 2, 4 cells, etc., morula. And then 9 months later, you have this. Okay? Fine. We care about from here to there, because everything is being formed, as you can see graphically in here. Well, what happened? This is something that I think would be very important to Swiss people, because this thing is a cretin. And the syndrome was discovered here because these mountains don't contain a lot of iodine. But now things are okay. So this cretin is aged 22 years and didn't have thyroid hormone during fetal development, so it looks like a toddler, and it has the body proportions of a toddler and the fat distribution. Okay, if you give thyroid hormones after birth, it won't go very far either, you see, so you lost it. So you have to act during fetal life not to lose IQ, for example. So this is an organizational effect. And that is what hormones will interfere with when you expose fetuses. This is the same problem, but in an adult person. So this adult person doesn't have thyroid hormone, or it has been treated with an antithyroid. And so the person is slow, has cold skin, has myxedema, dry skin, etc. You give this person thyroid hormone and it will be active again, it will be normal. Okay, so this is an activational effect of the same hormone. So that brings us to endocrine disruptors. What are those endocrine disruptors? Just exogenous chemicals or mixture of chemicals that interfere with any aspect of hormone action. And by that I mean the production, the disposal and degradation of hormones, as well as the activity of these hormones. Like, for example, it could be a hormone mimic or a hormone antagonist. that competes with the normal endogenous ligand for binding to the receptor, for example. And here are the familiar containers that we are talking about. So you can find endocrine disruptors in products like detergents, etc., but also in the lining of cans and in all sorts of packaging. And why do we worry about? Well, because of the fragile fetus, more specifically. And why we do that? Well, 25 years ago, Theo Colborn organized a conference, the Wainspread Conference, where Pete Myers and I were there 25 years ago. And we were trying to figure out what was going on with syndromes observed in the area of the Great Lakes in animals. And we use as model diethylstilbestrol. Why? This was given to pregnant women. This is a synthetic estrogen. The idea was to prevent miscarriage, but DES doesn't do that, actually. In 1971, when those girls were born from these mothers treated with DES, it was observed that very young women had clear cell carcinomas of the vagina. That is a cancer that is very rare and appears in women that are 60 and more. So that allows the identification of the syndrome. And that syndrome was described in humans. By the way, if you give DES to rats and mice, you obtain exactly the same syndrome. So if you happen to be a mammal and you're exposed to DES, you will get the same syndrome. So that we have to have in mind. If you're a mammal, you're going to have the syndrome. So what happens there? DES administered before the 13th week of gestation induces or provokes clear cell carcinoma of the vagina that manifests in young adults. It also produces anomalies of the oviduct and uterus and decreased reproductive success. It increases the risk of breast cancer at the age of prevalence, so that is when these women turn 50. And why is that? Now, this is the interesting thing. It's very interesting. For the epithelium of the uterus and vagina is pretty much the same until RUNX1 is expressed. That triggers a cascade of reactions that makes this cell, that is a cuboidal cell, to become a flat squamous cell. And that is the typical lining of the vagina. So here you have a stratified squamous epithelium. Here you have simple columnar. If DES is there, it blocks the expression of RUNX1, and so you will end up with the like islands of uterine epithelium inside the vagina. That is called adenosis, and this is the structure where cancer arises, clear cell carcinoma of the vagina. And this operates at a very specific age. I said that you have to be exposed before the end of the 13th week in order to have this problem. Now, this is not mutation, this is nothing like that. It's just that you didn't have the expression of a gene you needed at the right moment. Again, organizational effect. Now we get to a cousin of DS, that is bisphenol A, that is the building block of polycarbonate plastic, and it appears in many other things like epoxy resins that line cans, for example. It is produced in very high volume, and about not only you are exposed through food, but also transdermically, and also probably through the atmosphere in some cases, and dust, etc., since it's released in the atmosphere as well. So what is that we observe exposing animals to so-called safe levels of BPA? Early puberty. Some other groups observe changes in the prostate that lead to cancer. Altered mammary gland development, exactly like with DES. Changes in the uterus and ovary, exactly like in DES. Decreased fecundity and fertility, like in DES exposure. Changes now in brain steroid receptor levels and in behavior that are very much better characterized than in humans, all kinds of behaviors, that lead to believe that that the animals cannot fix attention and that they have the mouse equivalent of autism, predisposition to cancer, and obesity. So this is the syndrome you get if you expose fetuses to safe levels of bisphenol A. And now we'll see what happens, because, again, very complex. This is a transcriptome of the stroma of a mammary gland at fetal day 18. And here we see that bisphenol A is doing the same as ethinyl estradiol. And the pattern is very different than with vehicle. So this seems like, okay, just 5,000 genes that are modified with respect to the control. In a set of 30,000 genes, it seems to be something big. But if you look morphologically, what you observe is things that are very peculiar, like, for example, the stroma doesn't produce collagen. Well, that is important because of the biomechanics of ductal formation. Another protein present in the stroma, tenascin C, is not expressed. Fine, another problem for the interaction stroma epithelium. So that produces an accelerated maturation of the stroma, early development of adipocytes, and more growth of the epithelial tree. So this gland exposed to bisphenol A is bigger than this. Now, if it was all increased, it won't be a problem, but some of the processes were delayed. So some things got accelerated, some things got delayed, like the formation of lumen. And so we are in a process that starts abnormally. And then we see the animal at 1 month of age, when the animal is ceased to be exposed at birth. And we see there that there is a problem with the migration, the migration of the ducts. They are growing from here. Here would be the skin towards here. And if you put the control animal is doing very fine, it grew this far from the lymph node. This one is going very slowly. Then later on, and well, at the same time, we see that the problem is that they cannot make those ducts patent. Here there is an inhibition of cell death. This is cell death. It doesn't happen in the animals exposed in utero to bisphenol A. And then all of a sudden there is this induction of progesterone receptor forming clusters. Those clusters are the origin of branching. So by 4 months of age, this animal has a lot more branch and alveoli than the control. And you say, well, so what? I mean, the mammary glands, each time that you check them, diverge more and more and more. And then you see this that you never, ever observe in normal animals. That is the appearance of intraductal hyperplasias that are a preneoplastic lesion. So that appears only in the animals that were exposed perinatally. If you continue looking into this problem, you will see that this is, now I change animals, here is rats, so because it's a better model for breast cancer, for human breast cancer, so you get an increase of these intraductal hyperplasias as we saw in mice. And in addition, you see carcinomas in situ, which in women, and if you have a carcinoma in situ, you are operated. And if you wait a little later, you can see full-fledged tumors, carcinomas. So this recalls us, reminds us that women exposed to DS during fetal development develop breast cancer when they are 50. Animals exposed to DS do the same. Now, bisphenol A induces the same syndrome, so you can conclude that probably humans will have the same and are indeed having the same, but we cannot tell because we don't have a non-exposed population. This is very serious, I would say. And that brings me to the message hinted at by the title of my presentation. That is the difference between science for its own sake and applied science. I was saying here the time it took for us to figure out the first events on this DS induce clear cell carcinoma of the vagina. We are talking about 30 years of work. And the same about bisphenol A, we started in 1996, et cetera, et cetera. Meanwhile, people are being exposed, and more people are being exposed. So there is a problem there. And the problem is that basic science, science for its own sake, that is a lovely thing. I mean, the reason why I'm a scientist is because it's very nice to get to know how things work. There we live with uncertainty because there is always a new experiment to be done, always a T2 cross, always an I2 dot. And so we don't care about time. We care about how things happen. And this is the science of Galileo to Newton. It just took 100 years. But of course, the sun was always at the center of the planetary system and could care less about whether we knew it or not. So that's fine. Now, what about in medical practice? I just happen to be a physician. When you are in the emergency room and someone comes and has low blood pressure and is in shock, I don't open books and start reading and thinking, well, maybe we should do an experiment. You immediately take care of the pressure and all the symptoms and try to save a life with whatever you know today. And you are bound to do that. That is your commitment by the Hippocratic Oath. You have to act now. What about if you are a medical epidemiologist? Well, you are testing a pharmacological agent. How do you go about it? Well, you set your experiment such that it will favor the null hypothesis. That is, your default is nothing will happen. Your agent is not active. Why? Because if you are going to produce a pharmacological agent, you want it to be active. So you start from the default of the null hypothesis. Now, what happens if you are a public health officer or epidemiologist? Well, you are studying potentially harmful exposures. So your default has to be the alternative hypothesis. So you expect a deleterious effect, because you best err on the side of a false positive than a false negative. And so here we are facing a problem that we say is a potential problem, but maybe it's not a potential problem. It's a problem that is happening, but we cannot measure. That, like my rats, women are getting more breast cancer. And that, like my rats, children are more. There are more children expressing autism than before, and so forth and so on. And we just cannot wait until all the I's are dotted and all the T's cross in order to act. We have to do something now. It's our ethical duty. Thank you very much for your attention.